deoxyribonucleic acid, otherwise known as DNA, is held together with hydrogen bonds. A hydrogen bond is the intermolecular force occurring when a hydrogen atom that is bonded into a highly electronegative atom of one molecule is attracted to two unshared electrons of another molecule. It is composed of two long chains of repeating subunits called nucleotides. Nucleotide is a monomer of DNA. It consists of three parts: 5-carbon sugar, a phosphate group, and a nitrogen base. 5-carbon sugar in DNA nucleotide is called deoxyribose. The phosphate group consists of a phosphorus atom that is bonded to 4 oxygen atoms. The nitrogenous base consists of a nitrogen atoms and carbon atoms and is a base. How can only 4 nucleotides encode thousands of different genes? The sequence is almost like using the alphabet to spell different words. There are 4 bases: thymine, cytosine, adenine, and guanine. In DNA, cytosine only one strand pairs with guanine on the opposite strand, and adenine only pairs with thymine. These bases are called complementary base pairs. A replication fork is the Y-shaped region that results when 2 strands separate. As the replication fork moves along the original DNA, synthesis of one strand follows the movement of the replication fork. This then leaves gaps in the newly synthesized strand, which are later joined together by an enzyme called DNA ligase. Synthesis moves in opposite directions on complementary strands, like shown in the picture below. There are 3 steps to DNA replication. In step 1, enzymes separate the DNA strands. They move along the DNA molecule, breaking the hydrogen bonds between the complementary nitrogen bases. This is why the DNA strand is split in 2. The second step to DNA replication is DNA polymers. DNA polymers are where the enzymes make a new strand of DNA that is complementary to the original. That is why the yellow and green circles represent in the present photo. The third and final step of replication is the semiconservative final stage. In each new double helix, one strand is from the original molecule and one strand is new. It is called semiconservative replication because of each new of the DNA molecules has kept one of the original DNA strands. This process continues and continues until there are finally 2 new strands of DNA that are the exact same as the new one. A mutation is a change in the nucleotide sequence of a DNA molecule. When mistakes in DNA replication occur, that is when a mutation takes place. When mistakes in DNA replication occur, the basic sequence of the newly formed DNA differs from the sequence of the original DNA. When there is a mutation in the sequence, it can have a serious effect on the function of an important gene. The mutations that are not repaired can lead to several diseases, including cancer. Cancer can only occur if the mutations happen within genes that control cell growth and cell division, and therefore it is very important that mutations do not happen.